Hello there. I guess we'll start. Firstly, I have no idea why they put us in this room. I didn't ask for this room. Okay. You look nice there, sitting around the tables. It looks kind of sociable, but unfortunately, I don't really have any particular group exercises for you to do today. Okay, so it's just going to be a standard lecture format. Now, just to finish up with the last of the. Okay, thanks. So, just to finish up with the last slide from Tuesday, the last thing we really need to consider is the effect of radiation on bacteria, which, well, just like for you and me, can be extremely toxic. So, there's ionizing radiation from radio elements, which can give out alpha, beta, and gamma radiation, and then different forms of UV. Now, which way am I going to talk about this? Okay, yeah. So I'll maybe just go through each one here. So ionizing radiation, of course, can directly damage biomolecules, including DNA, but most of the content of a cell, yours, mine, or bacteria, is going to be water. So most of the time when you have, I don't know, a beta particle interacting with molecules inside the cell, it's going to be interacting with the water molecules. And this will form reactive oxygen intermediates, which are, chemical toxins, especially for DNA. So, a lot of the toxic effect is not directly due to the interaction between the radiation and biological molecules, but it's indirect. Now, before talking about resistance mechanisms, then it's very widely used as a means of sterilizing material that can't resist high temperature. Okay, so high temperature, 120 degrees in the autoclave for 20 or 30 minutes will destroy plastics. So you can sterilize lab culture plastics or plastics for medical use with a gamma ray source. So UV radiation, okay, you've got UV A, B, and C. So A, this is the closest to visible light, so it's the longest wavelength of different types of UV. UVC, well, I'll start with here really because this is the easiest one. Fortunately, we don't have to worry about it too much because it's blocked by the ozone layer. If we didn't have the ozone layer, we would not be able to survive on the surface of the Earth. And there are no real biological mechanisms of resistance to UVC because we're not subjected to it. But it can be used sometimes to sterilize water. So there is some kind of application of this because it's definitely going to be lethal for any kind of microbe. And I think that this is due to direct absorption by nucleotides. It's a direct effect. UVA and UVB are harmful but slightly less lethal than UVC. So UVA is strongly absorbed by tryptophan, so it's going to hit proteins and perhaps interfere with their function and their structure. So there's kind of effect via proteins here. UVB absorbed by DNA and can lead to the formation of thymidine dimers that have to be repaired. But once again, they can also exert a toxic effect through the formation of reactive oxygen intermediates. Now, there are some bacteria which are very resistant to ionizing radiation and some which are resistant to UVB. Now, the species that is well known for being resistant to ionizing radiation is Deinococcus radiodurans, which I better write out because my pronunciation is so bad. Yeah, so this is Deinococcus radiodurans. Now, this thing was kind of discovered during experiments to try and sterilize food with gamma radiation. And the people who did this at the end of the 1950s were able to culture some very radio-resistant bacteria, which turned out to be Deinococcus radiodurans. So this thing can really survive lethal doses of gamma radiation. And it seems to work because it's got a lot of enzymes that are involved in DNA repair. So it's very, very good at repairing DNA damage. Now, strangely enough, it doesn't. This bacterium is quite widely distributed. It doesn't only live in places with a high exposure to radioactivity. And it seems like the radio resistance is related to its adaptations to survive desiccation. Okay, so when the bacterial cell dries out, I guess DNA is going to be chemically damaged, and it's got a lot of repair mechanisms to survive and recover after periods of desiccation. So the radio resistance is kind of like cross resistance because the bacteria is selected and evolved to resist to some other kind of stress. And this also seems to be the mechanism of resistance to UVB. And this has been studied in photosynthetic bacteria that live in lakes above 3,000 meters of altitude in the Andes. So they survive and they grow well in a very high UV with a lot of UVB. So these bacteria that have been studied, what seems to occur is that the compatible solutes that these bacteria produce in order to resist the high salt conditions where they live in this lake also have an effect on inactivating and soaking up activated oxygen species. So once again, you've got kind of cross protection. So one type of resistance mechanism seems to help bacteria to survive UVB radiation. Okay, so that's about it. The only things I need to talk about. So just to summarize all of this. So for all of these environmental factors, temperature, oxygen, pH, salt, you have to have an idea of the definitions, be able to cite at least one bacterium, which is, halophile, acidophile, anaerobe, facultative anaerobe. You must be able to cite one bacterium at least for each category and have an understanding of the molecular mechanisms that are involved in controlling or resisting these extreme environmental conditions. Okay, so this afternoon, what we have to do is, before I forget, all right, so this evening we should have a microbe of the week. So I'll send out another question at 7 p.m. So be ready, be ready. So this afternoon we're going to be concentrating on energy metabolism in bacteria. And this is related to the kind of second interesting point about bacteria, is that they can live on practically anything, or sometimes almost nothing at all. And they can do this because they can perform a lot of unusual chemistry. So that's what we're going to be looking at this afternoon. Okay, so first, how do you, before you start metabolizing something, okay, you've got to actually internalize whatever it is that you want to eat. Okay, that's true for us. It's true for bacteria too. So we're going to start out with nutrient uptake. And then we're going to go on to, how to get energy from different types of molecule. And I think I'm going to talk about some basic points in physical chemistry first, just to make sure everybody is clear about these concepts. Yeah. And then once we've done that, then I'll go through different types of energy metabolism that occur in bacteria. Okay, so organoheterotrophs, getting energy from organic carbon. Firstly without oxygen, then with oxygen, and then we'll look at these weird types of metabolism, chemolithotrophs and phototrophs. So firstly, nutrient uptake. Now, there are not very many molecules that can freely diffuse across the bacterial cell wall. Small gases, okay, oxygen, carbon dioxide, methane, I guess, and water, of course. So most molecules that are going to be interesting for bacteria will need to get in the cell through some kind of membrane structure. Now, if the concentration outside the bacterium is high, then molecules can enter through facilitated diffusion. And that's particularly the case, for example, in the outer membrane of gram-negatives. So what you have is, or are, a number of pores in the outer membrane of the bacteria which just allow facilitated diffusion. So you'll have, I don't know, something like glucose here, which is outside the cell. And if the concentration on the exterior of the cell is higher than the concentration in the periplasm, then this glucose will just diffuse across the porin or the permease. So it's going to be present in the periplasm. And then once it's there, it's going to be transported into the cytosol by active transport, because inside the cell you have a relatively rich chemical environment. So the concentration of a lot of things which cells like, amino acids, phosphorus, sugars, can be higher than what you have out here. So in order for the facilitated diffusion to work, bacteria have got to reduce the concentration of all of the nutrients in the periplasm to like 0. So the concentration has got to be very, very low here, potentially very high inside the cell. That's why cells have got to put energy into this process to import glucose, amino acids, other stuff across the plasma membrane. So that's the way it is. So why do you have passive diffusion here and facilitated diffusion here and active transport here? Why don't they just have active transport in the outer cell membrane? Why is it difficult for bacteria? Yeah. Yeah, exactly. So this is permeable. You could lose ATP, for example. And it's the same as what we said for peptidoglycan synthesis, LPS synthesis. There's no energy out here to actually be able to run the active transport in the outer membrane. So that's why it must be organized like this and cannot be organized any other way for gram-negative bacteria. Okay, so these pores which mediate facilitated diffusion can be called permeases or porins. Okay, so permeases are substrate specific, whereas porins are just holes which will let through any small molecule smaller than a particular molecular weight. Okay, so porins and permeases allow small molecules to get into the periplasm, and then they're imported into the cytosol by active transport, and there are different types of transporters, which we're going to look at just now. Then the last thing we need to think about are specialized systems, particularly for iron and other metals. So ABC transporters. So this is for ATP binding cassette. So these are transmembrane protein. Well, okay, so the import system has two components. One is a soluble protein in the periplasm, and this is the one that will bind whatever needs to be imported. And then the complex between the solute binding protein and whatever needs to be imported will bind to the transmembrane pore, and the solute will be transported across the membrane, and this will cost ATP hydrolysis to put in the energy required for this. And this is very frequent for some sugars. Not the most frequent ones that bacteria are going to use first, okay? Arabinose, ribose, and then often for amino acids, glutamate, histidine, leucine. Another important molecule which is internalized by ABC transporters is phosphate. So this solute can be a small organic molecule or something inorganic. Okay, so this is going to cost ATP to import each of these solute molecules. Now the other way to import molecules across a membrane is by Symport Antiport system. So here we've got, inside the cell and the periplasm, and part of the energy metabolism of pretty much all bacteria is to maintain a proton gradient across this membrane. Proton and eventually potentially a sodium gradient. So that means you'll have a higher concentration of protons outside and a higher concentration of sodium outside. And this concentration gradient can be used to co-transport other molecules against the concentration gradient of, for example, sugar. So in this case, sodium is being transported together with the sugar, and transport down the sodium gradient is favorable, and that's what allows an unfavorable transport event to occur at the same time. So in E. coli, you have proton Symport to import lactose, some amino acids, but more often for these 4-carbon carbohydrates, malate oxaloacetate, okay, which have got a carboxylate group, so it's kind of easy to see where you can co-transport a proton together with these 4-carbon molecules. And sodium Symport apparently is used to import glutamate. Okay, so let's go back. So ABC cassette, arabinose, and amino acids like histidine, leucine, glutamate, proton Symport, oxaloacetate, and malate. Okay, yeah, I remembered them. Try and remember them. You need to know which import system is used for which type of substrate. And the last one is what's called group transport, also known as phosphoenolpyruvate sugar PhosphoTransferase system. And this is used for, well, not by all bacteria, but very, very often used for the import of glucose. So if you ever get this coming as a question in a test, What is the import mechanism for glucose? It's this one, group transport. Now, how does this work? Okay, what is happening here is that the import of the glucose across the membrane is catalyzed by this, what's called the enzyme 2 complex here. And the energy for the import is supplied by this phosphate group, which is attached to the protein subunit 2C. And the lysis, or the breaking this bond, this phosphate bond, provides enough energy to do two things. One, import the glucose, and two, transfer the phosphate group to the glucose to give you glucose 6-phosphate. Okay, so that's how the import is working. So where does the phosphorus come from? Okay, where does the phosphate group come from? It comes from phosphoenol pyruvate over here. So this is the last step in glycolysis, normally. Okay, so normally phosphoenol pyruvate is going to be dephosphorylated and give you pyruvate. And during glycolysis, this step will phosphorylate ADP to ATP. So normally you get one molecule of ATP from this reaction. But when group transport is occurring, the phosphate group first gets transferred to enzyme 1, then this is called the heat-stable protein, and then finally to the enzyme 2 complexes. And then it ends up on glucose 6-phosphate. Okay, so far so good. Now, the only other thing to say about this is, okay, so these subunits in the system, enzyme 1 and the heat-stable protein, these are the same, whatever the sugar is. But the enzyme 2 groups, they're going to be specific for particular sugars. So this is very common in many facultative anaerobes, some obligate anaerobes, and absent in most obligate aerobes. Okay, so why is that? Well, why are bacteria using this complicated system? Why don't they just import glucose with an ABC transporter, for example? Well, it's because of the energetic cost. So if you've got an ABC transporter, imagine that this is your alternative, okay? Transporting glucose. This will cost you ATP. Imagine you're going to have to spend one ATP molecule per glucose just to get it inside the cell. What do you want glucose for anyway? Yeah, you want to use it for respiration or your energy metabolism, whatever it is. And the next step is going to be to transform it into glucose 6-phosphate. This is the first step in glycolysis. So this would also cost you 1 ATP. So with an ABC transporter, you've spent 1 ATP to get the glucose inside the cell, and you spent 1 ATP to phosphorylate the glucose. And then, it's going to be used up in glycolysis. So this is the advantage of group transport, because you only need to spend 1 ATP and you perform 2 steps at the same time. You get the import and you get glucose 6-phosphate. So it's more efficient in energy terms. And as we'll see, anaerobic metabolism by fermentation does not produce a lot of ATP molecules per glucose. So if you can save 1 ATP, that's really important. Very important for anaerobes. For obligate aerobes, they're going to be performing respiration. And this will give you many more ATP molecules per glucose. So it's not such a big deal, you know, if you have to spend 2 at the beginning. Okay? So this is the important thing about group transport. It's complicated, but the cells, the bacteria that use this system really gain something in terms of their energy metabolism. Okay, so the last transport mechanism which you need to know about is for iron and for other oligoelements. And the reason it's difficult for bacteria to get iron is that ever since oxygen started to be produced by cyanobacteria about 2.5 billion years ago, oxygen has been precipitating out iron that's in solution. So iron 3, ferric iron, is very insoluble in the presence of oxygen. So the concentration of iron in most aquatic environments is very, very low. And that's why a specialized system is needed to pick up the really, really tiny concentrations of iron that exist. So in E. coli and pretty much every bacterium, then they do this by secreting what's called a siderophore. So a siderophore is a small organic molecule that has a very, very high affinity for iron 3, so for ferric iron. So these siderophores complex with iron. The affinity is so high that this can just pick up the very, very low concentrations of iron that are floating around, and then the complex is re-imported through an ABC transporter. So pretty much all bacteria have siderophores in order to import iron, and may also have other siderophores for other metals. One of the things about this is that the affinity of Enterobacter for iron 3 is so high that the formation of this complex is pretty much irreversible. So in order to remove the iron from this complex, then it has to be enzymatically degraded and taken apart, and then the iron can be used in some other important proteins in the bacteria. So siderophores, very important for picking up iron and other metals. Okay, so that's nutrient uptake. Now, before we look at particular types of bacterial metabolism, I always like to go back to basics a little bit about the physical chemistry of equilibrium reactions, and because the whole point of energy metabolism is to generate some kind of molecules that bacteria can use to get unfavorable chemical reactions to work. Okay, so whether a chemical reaction will go forward and take the substrates will combine to produce the products, or whether the substrates are just going to sit there and do nothing, depends on the Gibbs Free Energy, delta G of the reaction. This is what determines the equilibrium point of the reaction. And in the Gibbs Free Energy, you have, what's this? Delta H. Somebody said it, but it's so quiet I couldn't hear. Delta H is what? Enthalpy, yeah. Okay, heat, the heat release. And S is what? Delta S. Entropy. Right, good, okay. So what is important here? Okay, so it's minus T delta S, so the entropy term becomes stronger as temperature increases. Okay, good. So we all know this, that's very good. So the way that biochemically you can get unfavorable reactions to move forward is to couple an endergonic reaction, energetically unfavorable, to some other type of reaction which is highly favorable. So ATP degradation, release of the third phosphate group, will generate, about reasonable amount of energy, about 30 kilojoules per mole, or minus 30. So if you couple these two things together, then this reaction, which on its own would be highly unfavorable, can actually move forward. And don't forget that enzymes, they can accelerate the reaction, but they don't on their own change the overall free energy of the reaction. Okay, they can increase the reaction rate, but in order to change the overall free energy difference, then you have to couple the reaction with something else which is intrinsically favorable. So ATP hydrolysis is one which can be used to perform all sorts of useful tasks in the cell. Now, it's written ATP here, but it's not always ATP because, okay, when we were talking about peptidoglycan synthesis, what do we have? What were the monomers that first got formed? We had something N-acetylmuramic acid, right? Okay, was it ADP from ATP? No. It was uridine diphosphate bound to N-acetylmuramic acid. And the first step of to actually get this, you'll have the free sugar. Using up one molecule of UTP, so uridine triphosphate. And this is generally true, okay? So uridine triphosphates are used for the synthesis of polysaccharides. GTP, essential for the synthesis of proteins. It's GTP hydrolysis that powers the formation of the peptide bond by the ribosome. And CTP hydrolysis is used for the formation of fatty acids. So I don't know, I always like to talk about this because it just strikes me as amazing. You've got three different nucleotide triphosphates used for the synthesis of three different types of essential biological molecules. Okay, GTP, protein, CTP, lipids, UTP, polysaccharides. And ATP involved in a whole bunch of reactions. Okay, so all of these nucleotide triphosphates are used for pushing around unfavorable reactions. Often, okay, anabolic steps where you're building up a complex molecule. Okay, some kind of polymerization step is always going to be energetically unfavorable in a liquid environment, right? Because their condensation reactions, the concentration of water is so massively high that it's very difficult to push the reaction in the direction of condensation. Okay, next type of important type of chemical reaction that can supply metabolic energy. Redox reactions, okay? Electron transfer, oxidation, loss of electrons, reduction, gain of electrons. So every redox reaction can be broken down into two half reactions where something is going to give up electrons and this separation of charge is associated with something here which is called the standard redox potential of this half reaction. So these can be measured somehow, okay, in some electrochemistry lab. And so each kind of molecule that can exist in a reduced or an oxidized form can be classed as a good or bad electron donor or electron acceptor. So good electron donors have got negative redox potential. Okay, good electron acceptors have positive redox potential. And that means that electron transfer will occur from donors with a negative electron redox potential to acceptors with a positive redox potential. And so what you can do is you can calculate the kind of redox potential difference, I guess, which is also measured in volts, which is the redox potential of the acceptor minus the redox potential of the donor. So here we've got electrons being transferred from NADH onto oxygen. So you've got the redox potential of the acceptor, 0.82, minus the redox potential of the donor, which is minus 0.32. So that gives you a total of 1.14. Extremely favorable. Molecular oxygen, probably the best electron acceptor there is. So the redox potential difference can also be transformed into a delta G. Okay, don't really need to know this, but one of the things that's interesting here is the minus sign. Okay, so here we've got the redox potential difference is plus 1 volts, so that means it's a negative delta G, right? So it's favorable. So why is this useful? A lot of important biological molecules are reduced carbon compounds, like lipids, for example, okay? They need to be reduced. All autotrophs need to reduce carbon dioxide to fix carbon dioxide. So NADH, a good electron donor, is essential for a lot of bacterial and all cellular metabolism. So we need this to run or push redox reactions. And the last type of important physico-chemical energy for cells is the electrical potential across a membrane. So often a concentration gradient of protons, which is called the proton motive force. So bacteria will produce or will export protons into the periplasm to create a proton gradient, and that means that protons running back across the membrane is energetically favorable. Why is this energetically favorable, right? So this is associated with a negative delta G. You've got delta H and delta S. Which one is working here? Okay, I've got a proton, right? It's going to move from here to there. Is that going to liberate heat? Well, I would say no. It's just moving. It's not actually being chemically changed at all, this proton, right? However, the whole system is becoming less organized. Imagine if you've got like all the protons out here and 0 in here. This is the maximally organized state. Okay, entropy is the disorganization, the randomness in the system. So this, ions running down a concentration gradient is driven by entropy. Okay, so that's why it's favorable. And just so that we can get the formalism right, okay, so if you've got any kind of gradient of charge across a membrane, this can be related to a potential difference in volts by the Nernst equation. Now, the only reason I'm pointing this out is that what is important here is the ratio, okay? The absolute concentration of protons, one side and the other, is not really important. If you've got, I don't know, 1 molar out here and 0.9 molar here, it's not going to be very good, okay? If you've got 10 millimolar here and 10 micromolar inside, that's going to be much more effective. It's the ratio that is going to allow you to get a better, potential difference. If you've got a really good proton gradient, then that means you can power reactions that require, either a big redox potential difference or require a big delta G, like ATP formation. Okay, so what are proton gradients used for in cells? Okay, once we saw one example just now. So co-transport, import of malate oxaloacetate, flagellar motion to actually produce physical work of moving the flagellum around, and ATP synthesis. This is really essential for us. So in respiration, what is happening is you start with a redox reaction, oxidizing glucose to carbon dioxide, and at some point this allows you to generate a proton gradient, and this will give you ATP synthesis. So what are points to remember here? You've got 3 types of biochemical energy currency. ATP, reducing equivalents in NADH, and proton gradients. When spending them, using them up is highly exergonic. They liberate energy, and they can be interchanged. Generally via the proton gradient, okay? So the redox reaction can form a proton gradient when that can help you generate ATP, and it can also work the other way around. Okay, so they can be, if you can get one of these things, you can get the other 2, basically. Okay, so that's what we need to get, okay? That's what all bacteria need to generate from whatever it is they use as a source of metabolic energy. So we're going to kind of start from the simplest system, or the one that is perhaps most familiar anyway, is getting energy from organic carbon, let's say glucose, okay? And in the simplest case, this is going to occur without oxygen. So, firstly, we're going to look at what glycolysis is. So this is the steps from glucose to pyruvate. What are the 3 pathways that you can find in bacteria, and then what the problem is. If bacteria are just performing glycolysis, it's going to cause a big problem. And fermentation is the solution to that problem. So without really turning the whole thing into a hardcore biochemistry lecture, glycolysis is the following. So you start with glucose inside the cell. The first step is phosphorylation to give you glucose 6-phosphate. And it's transformed into fructose 6-phosphate, right? Then fructose 1,6-biphosphate, I believe. Okay? And so you go from just a plain sugar molecule to a sugar that has 2 phosphate groups on it. So you need to use 2 ATP to provide these phosphate groups. That's the first step. Then fructose 1,6-biphosphate is cleaved into 2 molecules of glyceraldehyde 3-phosphate. And the next step is where, I don't know, this is the next step is just like biochemical magic. So glyceraldehyde 3-phosphate dehydrogenase, GAPDH, this is the one that allows you to get something for nothing. Because what is going to happen is that this glyceraldehyde 3-phosphate is going to be what? It's going to be oxidized or reduced. That's strange. It's going to be oxidized, right? Because it's going to give up electrons onto NAD+. So the NAD is being reduced. That means this substrate is being oxidized. Gives up electrons. And this oxidation is highly favorable. Energetically highly favorable. And because it's so favorable, then this redox reaction can also be coupled to phosphorylation. And this gives you 1,3-biphosphoglycerate. And then afterwards, each of these phosphates are removed. And every time a phosphate is removed, this can generate ATP. So at the end of the day, once you have lysed 1 molecule of glucose to 2 molecules of pyruvate, you've generated 2 NADH and 2 molecules of ATP. Okay, because this is 3 carbon, 3 carbon, 3 carbon. You have twice as much here. So you generate 4 ATP at this step, where you had to spend 2 ATP in the beginning, so the net gain is +2. And it produces 2 molecules of pyruvate as a waste product, or let's say the end product of this pathway. So this is really the key enzyme that makes this happen is this one, okay, GAPDH. Now, the Embden-Meyerhof pathway, many bacteria have this, and this is what we have in our cells, okay? Also plants, yeah, all eukaryotes have this. But in prokaryotes, there are other glycolytic pathways that exist, okay? So these are the Embden-Meyerhof pathway and the pentose phosphate pathway. Now, I don't really think it's important for you to know the steps here. Okay, what is common are the steps from this aldehyde 3-phosphate. So this part here is conserved. And it has to be because this is the step and these are the steps that give you the energy. Okay, so these are conserved. What is different is, how you get the glyceraldehyde 3-phosphate from glucose. Now, the only one that's also worth mentioning here is the pentose phosphate pathways, because one of the first intermediates here is ribulo 5-phosphate. And this is important in producing ribose and phosphoribose as a precursor for nucleic acids. So pretty much every cell will have the enzymes of the pentose phosphate pathway. Now, if the enzymes of the Embden-Meyerhof pathway are also present, then the main pathway for producing energy will be this one. Whereas if these enzymes here are absent, then the pentose phosphate pathway will be doing everything, okay? Producing energy by glycolysis and producing ribose for nucleic acid synthesis. Okay, so what's the overall yield in terms of energy for the different glycolytic pathways? Okay, 2 or 3 NADH and 1 or 2 ATP. So from one point of view, you might think that's it, it's great. We've got 2 of the 3 things we need, right? And we can probably use one of these 2 to produce a proton gradient. And then our cell will work. But the problem is that in the energy economy of the cell, you need much more ATP than you need NADH. So the yield of glycolysis alone, in terms of ATP, is too low. So you might say, okay, well, that's not a problem. We'll just run more. We'll just eat more glucose. So we'll just metabolize more. We'll produce ATP. Great. But the problem is that won't work because all of these steps require a source of NAD+, okay? Oxidized nicotine adenine dinucleotide. Because if you convert all of your NAD+ to NADH, then this enzyme can't work anymore. And this is the step that produces the ATP. So you have to get rid of the excess electrons and regenerate the NAD+. And then you can carry on eating glucose and generating ATP. That will work. So that's the basic point of fermentation, okay, is to regenerate the oxidized NAD, and then the whole thing can run around and produce ATP. So you need to discard the excess electrons that are in NADH here. That is the real function of fermentation. Why bacteria don't just stop at pyruvate and throw out the pyruvate. So where are you going to throw these electrons anyway? Well, you know, you've got a whole bunch of pyruvate hanging around there. That's not going to be useful for anything else anyway. So pretty much all fermentation pathways are going to transfer the electrons onto from NADH onto pyruvate or one of its derivatives. So what is going to happen is that the reduced NADH can be recycled and reoxidized to NAD+, and then this can be used by the GAPDH step. So, you know, you put glucose in at the top, fermentation products come out at the bottom, the NADH is recycled, and it produces ATP. Bingo. We've done it. So that is the point of fermentation, is that so that glycolysis can just run around and around and around and produce ATP. So how does this occur? Okay. Well, there are multiple pathways of bacterial fermentation, but they all somehow start with pyruvate. Okay. And pyruvate here is the electron acceptor. Okay, where does the pyruvate come from? It comes from the glucose that was used at the start of the process. Okay, so in a sense, there's no kind of external electron acceptor. So once you've got the glucose, you produce pyruvate, and then you put the electrons on there. Okay, so let's have a little look at some simple fermentations. Easiest one perhaps is directly pyruvate to lactate by lactate dehydrogenase. Okay, so it's this ketone group here. It's going to be reduced to an alcohol, and the electrons for that come from NADH. It generates NAD+. So it's very common, used for making yogurt, of course. Okay. Lactobacillus, Streptococcus, Lactococcus, and fermentations, lactic acid fermentations can be homolactic or heterolactic. Homolactic, well, that means lactic acid or lactate is the only fermentation product. Heterolactic fermentations, the bacteria will produce lactate and something else. Okay. Well, what else can happen to pyruvate? Well, we can kick this carboxylate group out to give us carbon dioxide with pyruvate decarboxylase. This gives us acetaldehyde, and then acetal alcohol dehydrogenase here can reduce this ketone group to an alcohol again. So it gives us ethanol. So this is what yeast do. Okay, so baking bread, brewing beer, brewing wine, extremely important, very important process. Some bacteria can do this, but they are not important in the food industry. Okay, so there are, I think, some attempts made to engineer bacteria that can produce ethanol by fermentation, so that they can use things like, you know, cellulose and stuff like that as the carbon source. And then you could get ethanol production for, you know, biofuels. from, directly from plants, from cellulose. Don't know if it's really actually used as an industrial process yet, but this is kind of a potential use of ethanolic fermentation by bacteria. So Zymomonas is going to be the example to remember bacteria that performs ethanolic fermentation. So those are simple. They go from pyruvate pretty much directly to the product, to the fermentation product. Okay, so some fermentations are slightly less simple. So here we've got pyruvate. Or rather, 2 molecules of pyruvate being combined to give you alpha-acetolactate. So this is 2 by 3 carbon molecules. This is 5 carbon molecules. We've kicked out a CO2. And then this 5 carbon molecule is going to be reduced to a 4 carbon molecule. And then finally, you get the reduction step to an alcohol, to 2,3-butanediol. So once again, this is common. So Serratia and Enterobacter do use this one. Butanediol, the product here, is the precursor for the production of synthetic rubber. So it has an industrial use. And fermentation by Serratia used to be the industrial process to produce butanediol. Not really used so much now because the petrochemicals industry produces butanediol chemically. But it's a fermentation process that used to be important industrially. Now, for the other fermentation pathways that we're going to look at, you have to have an idea of what other metabolic pathways already exist in bacteria. And I mean metabolic pathways that use pyruvate. So in bacteria that can perform respiration, okay, then what is going to happen is that pyruvate is going to be converted to acetyl coenzyme A by pyruvate dehydrogenase. This is going to give another molecule of NADH, and then this feeds into the Krebs cycle, part of respiration. Another important use of pyruvate is to pick up another carbon to give you oxaloacetate, so 4 carbon molecule. This is a precursor for the biosynthesis of a lot of amino acids. So if you think about a facultative anaerobe, which will use oxygen if it's present, but can live in the absence of oxygen by fermentation. So it will have, the enzymes leading to this step here. Okay? And most bacteria will also have pyruvate going to oxaloacetate. So think about this because it makes some of the complicated looking fermentation pathways a little bit easier to understand. So propionic acid fermentation performed by Propionibacterium. Wow, I would never have guessed. So what is happening here? Okay, pyruvate transformed to oxaloacetate. Okay, that one kind of normal reaction. Many cells will perform this anyway. And then you have these other 4 carbon molecules all the way up to succinate, and then finally carbon dioxide is removed and gives you propionic acid. And once again, you have to use up NADH, so get rid of excess electrons to produce propionic acid. So this looks complicated, okay? Why are they doing this in such a complicated way? Lactic acid fermentation, you just need to have 1 extra enzyme. But here, they're using like I know 1, 2, 3, 4, like 5 enzymes here. Why are they wasting their time like this? Well, in fact, all of these enzymes here, they're already part of the Krebs cycle, except they can work the other way around. So that means, in fact, they're not adding 5 enzymes. They're adding 1 enzyme onto what they already have and what they probably will already use for aerobic respiration. So if bacteria are, using up all the oxygen, they've still got all the enzymes hanging around for aerobic respiration. They just need to add 1 on, and it will convert respiration to fermentation. So it's not so complicated, okay, if you think about the other reactions that are occurring in this bacterium at the same time. So, propionibacterium, propionic acid fermentation. And then another example of this is mixed acid fermentations, which are performed by many Enterobacteria. So, this is, I will admit, a little bit complicated, okay, because you don't have one product, you have a variety of different fermentation products. So, E. coli will do this. Now, what is happening here? So the first step is pyruvate getting converted to acetyl coenzyme A. So what normally happens is that this is catalyzed by pyruvate decarboxylase, and this will result in the formation of another molecule of NADH. Do we want this to happen if we're talking about fermentation? Let me just try and, so you start with pyruvate, and you're getting, okay, an acetyl group on coenzyme A, acetyl group. And this will spit out carbon dioxide. Well, actually, put the carbon dioxide this way. That's pyruvate decarboxylase performs this. So if you're trying to run, so if bacteria have got oxygen, not a problem. This is going to be oxidized by respiration, fine. If they don't have oxygen anymore, they want to get rid of the electrons on NADH by putting them onto some kind of pyruvate derivative. The last thing they want to do is generate any more NADH. Okay, this is just doing the opposite of what you want to happen. So in a mixed acid fermentation, it starts by a transformation of pyruvate acid to acetyl coenzyme A, but with a different enzyme. And this is called pyruvate formate lyase. So instead of oxidizing this pyruvate and reducing NAD+, what is thrown out as a product is not a highly oxidized form of carbon, that is CO2, but a slightly less oxidized form of carbon, which is formic acid. So you've got oh, that's terrible, isn't it? What a mess. Okay, so some of the electrons stay on the carbon here, okay, together with the hydrogen. So, pyruvate formate lyase gives you formic acid, and also acetyl coenzyme A. And then this acetyl coenzyme A can be transformed to a whole bunch of different products. Some of these other metabolic pathways can produce ATP. So that's interesting, all right, because the problem with glycolysis and fermentation is the ATP yield is too low. So this is complicated, but bacteria don't do complicated stuff for no reason, okay? And the reason that this is going to be useful is because some of these fermentation pathways can allow production of extra ATP. Okay? So that's good. So what you get when bacteria are undergoing or producing a mixed acid fermentation is you'll get a whole mix of fermentation products. Now, forget about this for a moment, but in alkaline conditions, you get ethanol, formic acid, acetic acid, lactic acid, all of these, different fermentation products. What they all have in common here, apart from the ethanol, is they are all acids. So these are going to be exported from the cell in quite large amounts, and the pH is going to go down. These bacteria are not acidophilic. They prefer neutral pH. So if the pH goes down too far, that will prevent these bacteria from growing. So when pH goes down to 6, what is going to happen is there's going to be a change. Not in all of these steps, but there'll be another enzyme formed here, which I think is called formate hydrogen lyase or something like that. And it's going to break down the formic acid into carbon dioxide and hydrogen gas. So as pH goes down, there will be production of another enzyme which will break down formic acid into hydrogen and CO2. And amazingly enough, this can be recombined to methane by other bacteria that live in the gut, by methanogens. Okay, what is the consequence of all this? Okay, so one of the consequences is the formation of methane by ruminants, cattle. Another one of the consequences here is the formation of some amounts of ethanol by normal gut bacteria. And this ethanol can diffuse into your bloodstream. So I think that's perhaps the reason why people don't have like zero ethanol at all in their serum, because your gut bacteria are always producing some, so there's a trace amount of ethanol in your blood. And this can actually be a problem for some people who have a very particular gut microbiome. And this was discovered because there was somebody in the States, right? I think it was a lady. So she was driving her car, kind of like all over the road like this, like going like this. The police stopped her and said, "Come on, blow into the bag, ma'am." So they gave her an alcohol test, and she was over the limit for alcohol. And she said, "No, look, I haven't had anything to drink all day. I'm telling you, I'm not lying." And it was true. She hadn't drunk any alcohol, and so she didn't want to lose her license or whatever. So she made a big deal of it, got the lawyer involved, and she was able to prove medically. She was able to show that she didn't drink any alcohol, and then medically it was found out that if she had a lot of sugar, this person, then the bacteria in her gut would produce enough alcohol to get her drunk. So it's kind of surprising, but so this is going on, in our guts maybe right now, but most people will just have trace amounts of ethanol produced. Okay, let's just go for this question, and then we'll have a break, I think, because it's really hot and I can see everybody is having trouble. So let's just do this and then we'll take a break. Okay, everybody okay for this? Yeah. Okay, which of these is a problem? Energy yield per carbon is low. Who says that? Nobody. Requires oxygen or another electron acceptor. That's a limitation of fermentation. No. Is anybody ready to answer any of these questions at all? Yes? Okay. Who's not ready? One or two. Okay, everyone else thinks they're ready. Okay, requires oxygen. Is that the limitation? Who says yes? Nobody. Only used by gram-positives. That's a limitation. Who says yes? No. Okay, nobody. End products can be toxic. That's a problem. Yeah, okay, good. Yeah, all these acid products are not good. Requires cytochromes. This is a no. Okay. Yeah, so you got one of them right. Okay, end products can be toxic. Okay, this is not true. Gram-positives and gram-negatives can perform fermentation. Obviously, this is not true. It occurs without oxygen. But this is a problem, okay? Energy yield per carbon is low in fermentation. So you need quite a lot of organic carbon available to live by fermentation. Think about it. You're getting like 2 molecules of ATP per glucose. Well, you know, you could live on that, but it's not exactly, you know, you're not exactly driving a Mercedes and staying in a 4-star hotel on 2 molecules of ATP. Okay. Right. So I suggest we take a break now. Okay, and then come back in 10 minutes or so and see if we can get some air in this room. Okay.